Well, I feel like I'm a warm-up band that is going off to the main show, so it's a bit sobering. I want to see if we can get things a little more grounded, but the question maybe doesn't sound like I'm going to do that. Quantum processing in the brain. Let me rephrase that as, are we a quantum computer? I mean, I think that's, in a nutshell, that's what Professor Penrose is really ultimately suggesting. That maybe we are. Now, there's a lot of skepticism, and I think it's a lot of it's perfectly legitimate, in fact. And why the skepticism? Well, basically, biological processes are slow. If you take a protein folding rate, it's a tenth of a millisecond, so 10 to the fourth hertz. Neuron spike firing frequencies, maybe 100 hertz. And if you take these processes which happen with the characteristic frequency, and you convert that frequency by multiplying by Planck's constant divided by Boltzmann constant to a temperature, you get a quantum crossover temperature, this T star. And for T less than T star, the system is cold. It's where quantum mechanics should hold sway. When temperature is larger than T star, you'd think thermal effects would swamp all of the quantum mechanics, and the quantum mechanics would be not manifest, if you will. Plugging numbers, a frequency of 10 to the fourth hertz corresponds to a quantum crossover temperature scale T star, which is about 10 to the minus 7 Kelvin. Obviously, orders and orders of magnitude smaller than body temperature, so one would conclude that biological processes are thermal, not quantum. The body is simply too hot for quantum effects, okay? But there's one loophole, and the loophole is that this assumes thermal equilibrium. And we know from the worldwide efforts to try to build quantum computers that the whole game is to isolate, to get them not thermally coupled to the environment. So isolation is absolutely paramount. And so let me ask, if we imagine that there could be some quantum processing going on in our brains, what could conceivably be the qubit that would serve as a sufficiently isolated quantum mechanical degree of freedom from the wet environment? You might say, could it be a microtubule? I don't think so at all. It's a very big thing. It flops around slowly. Could it be a protein? Those are big. Could it even be a water molecule? Well, they decohere very, very rapidly. Electron spins decohere rapidly. The only possibility for a putative neural qubit is nuclear spins. And nuclear spins can be very, very isolated. So let me just remind you, so a neutron and a proton have nuclear spin a half, and some atomic nuclei have nuclear spins. So for example, the sodium ion, if you take salt, sodium chloride, and dissolve it in water, the sodium ion and the chloride ion separate, and the sodium ion would float around, and you can do nuclear magnetic resonance and come in and measure the decoherence time, or the time it takes for the nuclear spin of the sodium ion to entangle with everything else. Now in NMR, there are times T1 and T2. Those times are equal to the decoherence time. Because the water molecules are fluctuating so rapidly, there's only one characteristic time scale. And that time scale is around a tenth of a second for the sodium nuclear spin. But that time depends on which nucleus one's looking at. If one looks at a lithium nucleus, lithium-7, that time is 2 orders of magnitude longer. That's 10 seconds. If one looks at lithium-6, a lithium-6 ion floating in water has a nuclear spin decoherence time of 5 minutes. And that is really quite remarkable. That's certainly a macroscopic time scale, on human time scales. But I think longer times are possible in this as well. So it's this very long nuclear spin decoherence time for lithium-6, together with some experiments on lithium isotopes fed to rats, which I'm not going to talk about here, which is what led me to contemplate the possibility, as surprising as it might be, that nuclear spins might be operational cognitively in the brain. Okay, so the way I've approached this is pretty straightforward. I've just listed a set of seemingly unrealizable conditions that would have to be satisfied by biology and chemistry and neuroscience in order for evolution to have had a path that it could have taken to evolve quantum processing cognitively. So here's this list of some of them. When we need a common biological element with a very isolated nuclear spin to serve as the neural qubit. What's the qubit? We need a mechanism for entangling nucleus spins. We need a mechanism for quantum processing. And we need a mechanism for quantum to biochemical transduction as well. And so, I sat down 4 or 5 years ago and started to try to answer these questions by basically saying, can such conditions be fulfilled in biology? And I'm sort of a typical theoretical physicist, so I know an enormous amount about next to nothing. But biologists and chemists know a lot about a lot, and so I sat down with my favorite collaborators, Wikipedia and Google, and had to learn some biology and chemistry to see whether these questions could be answered in the affirmative. So let me just take you very quickly through these. What about the neural qubit? Well, we need a common biological atom with a long nucleus spin entanglement time. And what that means, actually, is that we need a nucleus spin which has spin 1/2. And the reason for that is that nucleus spins 1/2 do not have electric quadrupole coupling constants, so that the electric field fluctuations, which in water are large, do not decohere the nucleus spin if the nucleus spin is spin 1/2. Moreover, we need that the element with the nucleus spin 1/2 to be in a small molecule that can tumble in water, so that one gets what's called motional narrowing, so that the magnetic fields fluctuate in effective direction. So here are the common biochemical elements: carbon, hydrogen, nitrogen, oxygen, phosphorus, and sulfur, and the common bioelectrical ions: sodium, potassium, calcium, chloride, and magnesium. Which of these have nucleus spin 1/2? That's the question. How many of them do? Besides the proton, which I'm not going to consider, there's just simply too many of them in water and everywhere else in biology, is phosphorus. So the phosphorus atom comes in one isotope, phosphorus 31, and that has nucleus spin 1/2. So in my view, the phosphorus 31 nucleus spin is the only possible neural qubit, and the phosphorus 31 nucleus spin may well not be being used for cognitive processing, and if it's not, we're not quantum computers. This is it. So what's nice so far is that we're trying to answer these conditions. What's the isolated common biological element that conserves a neural qubit? There's only one choice. So that's nice. There's no choice, in fact, phosphorus. Where does phosphorus occur? Well, phosphorus does it occur in a small molecule? Ideally, it would be a small molecule in which the other nucleus spins inside the molecule, other atoms inside the molecule don't have any nucleus spin. Well, phosphorus basically always occurs in a phosphate ion. Adenine triphosphate, adenine diphosphate, phospholipids. And that's a phosphorus atom with a tetrahedral arrangement of oxygen ions surrounding it. The oxygen ions have no nucleus spin, so that's nice, and it's this nice tetrahedral cage protecting it. How did I see? I know how to use up these things. I basically break, like classical electronic equipment is not my thing here. So anyway, so the phosphate ion is nominal charge of 3 minus. Now you can take a phosphate ion, have it floating in water, and use NMR to do phosphorus 31 nuclear magnetic resonance to measure the decoherence time of the phosphorus nucleus spin. And one finds a pretty modest time, not that long, about a second or so. And the reason it's not significantly longer than that is that at physiological pH, near 7, protons, which are freely floating around in the water, can bind to these negatively charged oxygens of the phosphate ion. And then once they're bound, the proton nucleus spin and the phosphorus nucleus spin start talking to one another. They interact with one another. They couple to one another. And that would be okay. But what's not okay is that the proton is hopping on and off into the water, onto the phosphate, off again. And so then, as soon as that happens, the game's over, okay? So this doesn't look very encouraging, but I wasn't about to give up, because I'm trying to do some reverse engineering, and one has to keep an open mind for that. So basically I asked, can there be another positively charged ion, another cation ion, which could outcompete the proton in binding to the negatively charged oxygens of the phosphate ion? So you can ask, what about the sodium plus, potassium plus? As far as I know, they don't take any part in chemistry, in biochemistry. What about magnesium 2 plus? Well, magnesium 2 plus cannot outcompete the proton in binding to the phosphates unless the pH is around 10 or 11, in which case you get magnesium phosphate precipitating out. That leaves just calcium 2 plus, and calcium 2 plus appears to do the job. The hint that this maybe can happen is that calcium phosphate is bone. Bone is a mineral, a crystal. It's hydroxyapatite. Now, in 1975, a man called Posner was looking at the X-ray crystal structure of bone, and he noticed that inside the unit cell, it's a big unit cell, there were these two structural units, calcium 9 phosphate 6, which is schematically drawn here, two of these so-called now Posner clusters, which were in the unit cell, and that was then repeated in the bone crystal hydroxyapatite. So this is like a 1 nanometer diameter. Now fast forward some 35 years, and there's a number of experiments which have found reported evidence that these Posner clusters actually can exist as stable free-floating molecules in simulated body fluids. Now, if that's indeed the case, this is very, very nice because inside these Posner molecules, the oxygen nuclear spins don't exist. The oxygen has no nuclear spin. The calciums have no nuclear spin. So the only nuclear spins inside this almost spherical molecule are the 6 phosphorus nuclear spins. And so one expects, moreover, this is almost a spherical molecule, so it can tumble very rapidly. So one expects very long phosphorus 31 coherence times for the Posner molecule in water. Peter Hoare, who's one of the advisors, I guess, to this conference, did some calculations trying to estimate these decoherence times, and he came up with 37 minutes. I think it's much longer than that for reasons I can explain in private. I mean, I think he assumed some things which are not valid. But anyway, even with that, very conservative estimate, 37 minutes is quite long. So the Posner molecule appears to be ideal for quantum storage and maybe quantum processing. Now, where might Posner molecules occur in vivo? The only place that they would occur, as far as I can tell, is in mitochondria. Now, of course, we all know about mitochondria as the place where ATP production goes on, but mitochondria are also, another role is that they sequester and release calcium in a concerted, in a way that's used physiologically inside a cell. Now, inside the mitochondria, the calcium is actually stored in calcium phosphate, some sort of calcium phosphate sludge. And that's where we believe that these Posner molecules might well, or presumably do, exist. Another nice thing about mitochondria, which is maybe slightly less well known than the ATP production, is that mitochondria are not just some stationary organelle, that they are continually breaking apart, they're fissioning, they're fusing together, they're moving up and down microtubules. So the conjecture here with the Posner molecule formation and melting inside mitochondria, entangling non-locally in mitochondrial networks via fission and fusion might be conceivably underlying some putative quantum processing with nuclear spins. Let me just see if I can run a video here. This is just off YouTube. What's shown here is mitochondria, green and red there, moving on microtubules. They can fuse together, fission. They move about a micron a second. That's as quick as they can move. You can see them sped up. And let me show you one other video. Maybe those in the front that can see this. It will look like a bunch of chicken wire with little insects crawling on them. I don't know if you can actually see them. You probably have to get right up at the front. So it's quite an elaborate dance of mitochondria moving up and down these microtubules. Presumably it has some function. And the function it could have in this story would be to lead to non-local entanglement when a mitochondria fissions, and then when they move around and release calcium to transfer the entanglement into a biochemical activity. So how am I for time? Maybe what do I have? 10 minutes, or who's keeping track? Okay, I'll try to go through this very quickly. So what about quantum processing? How could one have quantum processing with Posner molecules? So there's sort of four aspects of this. There's a nuclear spin entanglement, which entangles with the rotations of the Posner molecule. There's something called quantum dynamical selection, some Posner pair binding, and then some quantum information with circuits and projective measurements. There are 6 nuclear spins inside a Posner molecule. They interact with one another through this exchange interaction. What's important for what I want to say next is that these Posner molecules have a rotational symmetry. There's a threefold symmetry axis around which, if you rotate the molecule by 120 degrees, it comes back to itself. And because of that, you can label the 64 nucleus spin states, 2 to the 6 is 64, 64 nucleus spin states, by a pseudospin tau, which takes on 3 values, 0 and plus or minus 1, which tells one how the nucleus spin states transform under rotation by 120 degrees. And they transform as the cube root of unity may goes to e to the I2 pi over 3 times raised to the power tau. So these three pseudospins, a bit like the ortho and para states of molecular hydrogen, but here, because there's a threefold rotational symmetry, there's actually 3 of these, I'll call them isomers, if you will. And just like with ortho and para hydrogen, where the nucleus spin states and the singlet and triplet nucleus spin states are entangled with the rotational states of the molecule, likewise here for the Posner molecule, this pseudospin is going to be entangled to the orbital angular momentum L, where here L takes on any integer. It's the orbital angular momentum around the one symmetry axis. And L is going to have to be equal to tau plus 3 times an integer. And so the total wave function is a sum over the 3 pseudospin sectors. And the pseudospin, each sector is a linear combination of these plane waves, or these angular momentum eigenstates, with angular momentum L equals tau plus 3 times z. The typical thermal angular momentum of a Posner molecule, just based on its moment of inertia, is around 1,000 in units of h bar. And so what you're supposed to think about is that these 3 sectors, these 3 pseudospin sectors, have angular momentum, typical angular momentum, let's say 1,000, 1,003, 1,006. That's one sector. Another sector would be 1,001, 1,004, 1,007. That's a second sector. The other one, I guess, would be 1,002, 1,005, 1,008. So this is this sort of meshing that it's very, very difficult, in fact, for the environment to measure this pseudospin sector. Let me call it a Q-trit. It's basically a three-dimensional Hilbert space, which is extremely isolated. It's much more isolated than the nucleus spin, than the total nucleus spin itself. And what's more amazing, really, is that if you look at these different pseudospin sectors, it's only one of the pseudospin sectors, tau equals 0, for which you can write down a wave function which is real and not rotating. Now, with a single quantum particle which is moving in one dimension, you might say, how do we know when that particle is not moving? Well, one way to stop it moving is put it in a box. When you put it in a box, it has a standing wave. The wave function is real. Likewise, for rotational angular momentum, if you want to say a molecule is not rotating, you need a real wave function. But for these 2 pseudospin sectors, tau of plus or minus 1, you cannot write down a wave function which is real. And so those pseudospin sectors are always not rotating. They're always rotating. So the real non-rotating wave function is only possible when tau is equal to 0. And then there comes this something called the quantum dynamical selection rule, and this is where we conjecture there's a wave function collapse. So a bit similar to what Sir Roger was telling us about, but it's not with gravity. So imagine this rotating Posner molecule, which is trying to chemically bond onto perhaps an amorphous calcium phosphate a substrate, if you will. It's just really for sake of illustration. So it's rotating around. If it chemically bonds, it stops rotating. Well, we've just seen that a real non-rotating wave function is only possible when tau is equal to 0. So we then conjecture that chemical binding of this rotating molecule implements a projective measurement onto tau equals 0. And this is a projective measurement. Professor Penrose talks about the 2 aspects of quantum mechanics as unitary time evolution, and then there's a projective measurement. We don't really understand what projective measurements are, except we make them in the lab. We're conjecturing that the projective measurement is being made right here. When one has these Posner molecules chemically bonding onto these stationary substrates. But what's more interesting is the binding of 2 Posner molecules. You can do density functional theory calculations and show that, at least in vacuum, energetically, it's favorable to have these 2 Posner molecules chemically bond together. if two Posner molecules come together and they're tumbling on one another and they chemically bond, they stop rotating, and that induces, we conjecture, a projective measurement onto a pseudospin singlet, a case with tau A plus tau B is equal to 0. And this gives one a way to quantum entangle the pseudospin of Posner molecules. Imagine two Posner molecules approaching one another. Situation one is that let's say they chemically bond to one another. They stop rotating. That makes a projective measurement onto the pseudospin, total pseudospin singlet. Then imagine that they separate. They become entangled that way. The pseudospin becomes entangled. Alternatively, let's imagine they come together, they try to chemically bond, and they don't. Then that implements a projective measurement onto 1 minus PS. So then they become entangled. So either way, when two Posner molecules come together and attempt to chemically bond with one another, and either able to or not, that implements a projective measurement which entangles those two Posner molecules, entangles the pseudospin of those two Posner molecules. So that's how one could get entanglement. And quantum processing with Posner molecules, one would have networks of pseudospin entangled Posner molecules. You should visualize this inside the mitochondria. If this pseudospin entanglement really is present, it will influence non-locally further pair bonding or pair binding of Posner molecules. So for example, if A and A prime, two Posner molecules have chemically bonded together and come apart, and B prime have chemically bonded together and come apart, so they've each been projected onto the pseudospin singlet. If A and B later chemically bond to one another, then A prime and B prime get projected onto the pseudospin singlet, even though they haven't hit one another yet. If they do, the probability that they chemically bond will be 3 times higher than if A and B hadn't chemically bonded together. So you would possibly have, non-local entanglement between chemical reactions. Here the reaction being the chemical bonding of two Posner molecules. An absolutely crucial feature which one would need to have if the story were to be complete would be a quantum to biochemical transduction. There must be some way to have all this entanglement, if it's there, and Posner molecules to be transduced into chemical reactions and biochemistry, basically, action potentials running down neurons and so forth. Well, one can argue that a Posner molecule tumbling around is going to be pretty stable to protons trying to dissociate it. But if two Posner molecules bond together, they're going to rotate more slowly, and under the right pH conditions inside the mitochondria, slightly more acidic conditions, at least than outside the cell, these Posner molecules a pair of Posner molecules could dissociate. That's a chemical reaction. They could dissociate into their constituent calcium and phosphate ions. And once you have free calcium ions, and if they're released from the mitochondria, those calcium ions can, if you're in the presynaptic neuron, for example, can trigger neurotransmitter release very effectively. So the idea would be that released calcium ions modulate the neural activity, creating some sort of quantum network that coexists with the neural network. So let me just start wrapping up here. I mean, basically, here's the classical brain. And by classical, I don't only mean non-quantum, I mean all. It's the field of neuroscience is classical. The only quantum neuroscience you'll hear about is maybe Roger Penrose and me. Maybe there's 1 or 2 other people who are. But what I've been talking about is a substrata of Posner molecules getting entangled in mitochondria and speaking to the neurotransmitter release and so forth in such a way that it has to be very intricately connected. So what you're supposed to be thinking of is basically this is like the quantum computer, and all the rest of our brain, which is classical, is like the experimentalist. That's the classical computer, right? Our brains are classical, let's say, completely. Well, they're pretty amazing at processing, right? And so that's now if you have this substrata, this quantum substrata, the classical brain, our classical brain is helping guide that. Is doing what? I mean, one has to have chemical reactions, have to implement projective measurements on the nuclear spins, and the nuclear spins have to modulate chemical reaction rates in some way, and that's what I've been talking about. So the story here is obviously just partly formed at best a picture puzzle. What's needed are experiments to try discerning the precise shapes in the various pieces of this puzzle and see if they actually start fitting together. After 2 years of effort, I managed to get some funding to do some experiments thanks to the Heising-Simons Foundation. And we have 4, not so well-funded efforts, but all under the umbrella of the Heising-Simons Foundation to look at quantum brain motivated type experiments. One is looking at lithium isotope dependencies on rat behavior. The third project is looking at these Posner molecules and trying to, for example, do phosphorus NMR and measure the decoherence times for the phosphorus nuclear spins. We've got some experiments planned in mitochondria. I haven't mentioned here the role that pyrophosphate and pyrophosphatase enzyme could be playing as well. And we have some experiments on that. Okay, so let me just wrap up. So I told this story to a brilliant theoretical physics colleague of mine in Santa Barbara. And he said, well, that's all fine and well, Matthew, but he didn't think evolution was clever enough to have figured out a path to find a way to evolve a quantum computer. And I thought, So I said to him, well, careful, because evolution was clever enough to evolve you. And he had to admit he was pretty impressed with that. But then I started thinking a little bit more about it, evolution and quantum computing, right? So let me term this, when will we have a multi-purpose quantum computer? So let me frame it this way. Now the recent path towards a quantum computer is pretty short. 1982, Richard Feynman suggested that we might have reasons to try to process on a quantum machine. Peter Shor in 1994 had, of course, his algorithm for factorization, prime factorization on a quantum computer. And in the last 10 years, I mean, the big companies are spending, $1 billion a year collectively, trying to build quantum computers. So, it's an enormous effort. But I want to take a longer view towards a quantum computer. So let's imagine that it took, as it did, 3.5 billion years for life to evolve and become primates. And 3.5 billion years later, there were human beings who were very smart. Maybe they were just classical computers. Very smart, though, and they worked at Google, and they were helping trying to build superconducting qubits. Okay? Well, if they're successful in building superconducting qubits and building a quantum computer, evolution evidently found a route to build a quantum computer through these clever classical Google engineers. Think about it, it's true, right? I should have said here, so multi-purpose quantum computer, I'm going to say 50 years. I'm a bit of a pessimist, but maybe it's 10 years, maybe it's 5, 50 years. Okay, so now let me just ask the question, how many years will it take for evolution to design or build a quantum computer? 3.5 billion years plus 50 years? Or just 3.5 billion years? And I don't know. I think the second one's not crazy. I think it's a lot longer effort and time. And so let me just finish with acknowledgement to Heising-Simons. There's been several theorists that are involved, a number of experimentalists who are getting involved. And thanks for your attention.
Thank you very much. Can I ask a quick but probably naive question? Wouldn't NMR wipe your brain clean?
No.
Why not?
It wouldn't. Well, for one thing, this pseudo-spin that I was talking about is blind to a magnetic field.
Okay.
Yeah.
Okay. Good answer. Yeah.
So do you have any notion of a calculation that would be done, or it will be done with this spin system coupled to calcium and neurons, and that would somehow motivate why this would be a good thing for the brain to be doing?
Yeah, okay, so we're starting to look at quantum circuits.
Yeah, that's what
Yeah, and it's actually, it's very natural. In fact, if you take the basic model that I've considered where they can chemically bond together to make a projective measurement, adding one extra ingredient, which is if they bonded to one another, they distort, and there's a unitary time evolution which mixes the different pseudospin sectors, and then they come apart. If you take many such Posner molecules and you run that circuit, what you find is that it evolves into a cat state where you have a linear superposition of, let's say, all of the Posner molecules in the tau equals +1 state, plus all the poser molecules in the tau equals -1 state. Now you're supposed to imagine that that's some sort of cat, and then that fissions mitochondria, and then you have two entangled cats, and then you have another two entangled cats, and you start bringing these entangled cats together, and you get you can get a situation where when you bring this pair of entangled cats together and this pair of entangled cats together, you get a lot of chemical bonding between the poser molecules with one another, and perhaps in sync release of calcium out of those mitochondria, even if they're non-locally located at that stage. So people ask me, is this a universal quantum computer? Do we have enough gates? No, at this stage we don't. People also ask me, what good would it be if we have quantum computing in the brain? Maybe awareness would be a nice side effect of it. I don't know.
So, so you said that the spins are invisible for external field.
Well, the pseudospin is invisible.
Right. I mean, so
Right.
but it still will polarize real spins if you put somebody in an MRI
Well,
machine.
actually, it depends on how long the nuclear spin decoherence times are. If they're 37 minutes, it would take a magnetic field of 37 minutes to polarize, and it doesn't polarize very much.
But
right?
I'm saying that
it's only one part in 1,000.
we've been putting people in MRI machines for almost 50 years,
And
there is no
yes.
evidence that there is any cognitive
Well, you're wrong.
deficiency.
Have you ever tried? Okay, excuse me.
I'm sorry.
Yeah, thank you.
Yeah.
People are wheeled slowly into the fMRI machines for a reason. If you go and put your head near the bore of the magnet, it's about 1 or 2 Teslas, you take your head, you shake it like that. After about 10 seconds, you get dizzy. This is something that NMR experimentalists have told me that they know the stray fields in an NMR magnet. You put your head through, and you feel it. So we can actually detect magnetic fields with our brain. Now, it could be eddy currents, conceivably, or it could be something, coupling to the nuclear spin.
So I have stayed.
Can we move
I just want to say
question.
something, please.
Okay.
I'm so sorry.
But we have to have other people having
I understand that. That's why we have time for discussions, right?
Well, we're already into...
I mean, let's not worry about it, okay? But so I have done experiments on me, 3 hours in the MRI machine at 5 Tesla. And we look at the consequence. And so I can assure you that these are eddy currents. I mean, at least what we know. I mean, okay, it's possible that perhaps there is something that is quantum.
Well, we know other effects of magnetic field. There's something called transcranial magnetostimulation, which is actually used to treat depression, right?
But that's because these are polarization of the membranes. I mean,
Well, they may be eddy currents again. We should be able to test that actually, but that's something that hasn't been done. But,
On the back, please.
I'm just, but perhaps a minor technical question, but I think it's of general significance. You regard the coupling of these molecules to a substrate as a projective measurement onto tau equals 0.
yeah.
But on the other hand, the system is no longer, after this binding, this is no longer a Posner molecule in tau equals 0. This is now a bound Posner molecule to a substrate. That's quite different than a
No,
projective
exactly.
measurement.
But the ones that try to bond to the substrate and are unsuccessful, they get projected onto tau equals +1 and tau equals -1. So they actually become a qubit. So and once they become that qubit, then they can't bond to the substrate anymore because they don't have any pseudospin tau equals 0 sector.
So it's serving as a filter. Is what
So
you're saying.
if some amorphous calcium phosphate is there, which it presumably will be, it will serve as a filter and to do two things. One is to project the Q trit down into a qubit, and the other is to stop those other Posner molecules from actually bonding to the substrate and getting precipitation. I mean, you don't want bone growing inside the mitochondria, right?
Question, please.
Hi. So this mechanism would then be operational also in other animals with neurons, right?
Would it be operational in other animals?
It should be.
Well, singular cellular organisms don't have mitochondria.
Sure.
Okay.
But they have to have neurons.
You presumably would have to have neurons. Now, the calcium and phosphate, I mean, you want vertebrates good because, bone is calcium phosphate, and you want calcium and phosphate regulation being, really is important. The other thing is mammals, mammalian milk is basically a way to deliver liquid bone. I mean, you've heard that milk, the calcium is good for your bones, right? When you drink mother's milk, well, it's because milk is delivering calcium phosphate. So, if we can actually understand these things, I mean, if quantum processing really is present cognitively, we should be able to manipulate it and change it in a way that's predictable, and then we can try it on your dog or something, and then see.
Well, we've got to go into the microphone for the recording.
Maybe I didn't get it right, but is there some subtle assumption about the nuclear spins? Because you started from a long-lived nuclear spins, and then you moved to pseudospin. Now, to have qubits with pseudospins, etc., does it require to have a pure state of nuclear spins?
To begin with.
To begin with?
Yeah, well, that's a very good question. And, that's like when you do a quantum computing experiment, there's the state preparation, right? And the state preparation has to be a disentangling of the qubit from the environment. And so one way that you might get that disentangling, which I didn't have time to talk about, is when adenine triphosphate breaks down into adenine monophosphate, which it can, you get two phosphates which are tumbling around one another, and their nuclear spins are entangled with one another predominantly. And then there's an enzyme pyrophosphatase, which is there inside the mitochondria as well as in the cytoplasm, which breaks these two phosphates apart, and then the nuclear spins of those two phosphates are predominantly entangled with one another. And so if then they get encapsulated in a Posner molecule, you're sort of disentangled from the environment. So, but that's a very, very good question. Is when does a disentangling take place, if it does? So thanks, that's a good.
Yeah, I had a question for the other end of the story, which is, so the fissioning and fusioning of mitochondria is a regular biology process, which means there are genes that control it. The rates are probably different in different people.
Yeah.
So have you looked at all about, I mean, just the simplest thing of correlating people's intelligence, measure however you want to do
but there's
it
a lot
with genetic diseases that modify the rates?
I'm not an expert, but you can read that the correlates with mitochondrial dysfunction and Alzheimer's, for example. Now, does that does that help? There's correlates with a lot of things, with a lot of other things.
With a lot of
exactly.
Alzheimer's is such a broad spectrum of things that go wrong that it's hard to know which is the which is the.
I mean,
the
but there's
forest,
many, thousands of biologists who are working on mitochondrial specialists.
Sure, I'm just wondering if there's some story there that somehow, this way of thinking about it somehow sheds light on a story which otherwise the biologists have found completely, incomprehensible. Like, why do you get these fissions? What happens if you knock out some proteins that are essential for that process?
But that's not the quantum physics that we're looking at, right?
I know, but we're trying to figure out. I mean, the rest of us are trying to figure out some, probability that this is worth thinking about from our perspective. So any evidence in favor of that would be helpful. So I'm just wondering if there's any biological evidence in favor of that rather than.
There's no direct biological evidence in favor of this. No.
Judith has a question. Could you pass the microphone up?
Just a quick question. You said something really interesting, that the disentanglement could involve ATP undergoing a series of hydrolysis to AMP and then pyrophosphatase. But what happens when you hydrolyze ATP is you order the pKa's of those phosphates, and they're going to take up protons rather rapidly, which would then give you nuclear spin interactions, which would destroy the uniqueness of the phosphorus spin. So I wonder if you could just comment on that.
Well, okay, so what I'd be thinking of is that pyrophosphate, which is diphosphate, let's say goes through pyrophosphatase and breaks into two phosphates. You'd have 1 second before those phosphates would have to be encapsulated in these calcium phosphate molecules in order to protect them from the protons.
So there has to be a competitive mechanism.
Yeah, but a second is a long time, right? I mean, if this is happening, a mitochondria sequesters calcium and there's phosphates being released, then it can happen quickly.
Right, but I think the enzymes may be operating in milieus which are fairly distant from calcium. I have no idea about the organization.
Well, no, but there is a mitochondrial pyrophosphatase.
Yeah, sure, but I just don't know in the organization.
I'm not sure exactly. either. I mean, it's in the matrix, but
Could I just ask one quick question? Well, I hope it's going to be quick. Why do you think quantum mechanics is involved in consciousness? Is there a simple
no,
way
I
to
don't. I
explain
think
that?
it's an answerable question. That's what I think. And so, do I believe that the story I'm telling you is correct? No. Do I believe it's a story that we can try to check to see whether it is correct? I hope so. Otherwise, I am really wasting a lot of time. But my original motivation was to try to understand lithium and these remarkable experiments feeding lithium isotopes to rats, where the lithium-6 isotope had a completely different sign of the effect from the lithium-7 isotope. And this was really striking. This is what got me involved in this in the first place. I didn't start out saying I'm going to look at quantum processing in the brain. I started out trying to understand why lithium is such an effective drug at bipolar disorder.
Okay, thank you. Now I believe we have an announcement.
Yeah, I wanted to attract your attention to a potential funding opportunity. You know that NSF has 10, is this on? Yeah. Okay. 10 big ideas, and one of them is Quantum Leap. And recently we were invited to join, so we joined to the Quantum Leap working group. They were in the middle of preparing second quantum incubator solicitation, so we were able to put in biology as one of the acceptable areas. And the solicitation came out just about a week ago or so. And the only requirement is basically proposals should be multidisciplinary, so biologist, physicist, engineer, chemist, at least 2-3 groups needs to be joined to apply. So there's a letter of intent, and then there will be pre-proposal and the full proposal session. So I'm going to send you the link after the meeting. So I would appreciate if you forward it to your friends or yourself apply. This is the most suitable community to announce this.
Yeah, thank you.
Can I ask
Thank
a
you
quick question
yeah.
about that? Is funding available outside for groups outside the US?
No, NSF doesn't do that, but the collaboration still is acceptable.
Okay. It's interesting we had a quantum technology funding round in the UK. They excluded biology specifically from it.